Hello students, welcome to LEELA'S tutorial. So today we will discuss about mouse models for cancer research. Why we have to use the mouse models for cancer research? Because these mouse models have high mimic to the diseases which are obtained genetically in humans. So that it is very easy for the study of cancer which is occurring in humans and how it can be cured. Right? So how the mouse models get respond to the drugs which are used for curing the cancer. So here the mouse models are genetically modified or genetically altered to overexpress or lack specific genes such that they either induce or inhibit tumor growth. Right? So what you are doing, first you are selecting the mouse, then you are standardizing the mouse. After that, you are either inducing by using carcinogenic agents so that the mouse gets cancer. Then you go for the study how the cancer is progressing in that mouse, in which region and what is the gene responsible for causing that cancer. Right? And in another way, here it is also used in studies of inhibition of the tumor growth or cancer. That means how the cancer can be treated using the certain novel drugs and what is the response of that mouse to that drugs can be studied. So main thing is why the mouse models are used, because they are highly standardized inbreed strains which mimic the same diseases which are produced in humans also. Right? And here you have the tumors. The mouse have the tumors which resemble at genetic and morphological levels. Right? So here as they are resembling the humans at genetic and morphological levels, the tumors or the cancers can be studied easily by using these mouse models. So apart from this, first we should know what is difference between cancer and the tumor. Right? So cancer is a disease where cells divide continuously. Right? They divide continuously and gradually they cause destruction of the tissues. Right? Whereas the tumors, they may be dividing continuously, but all tumors are not cancers. The tumor can be a group of cells which may be a cancerous cell or it can be non-cancerous cells. Right? If it is a cancerous cell, it will spread to the other parts of the body which is referred as malignant tumors. Right? So now let us see what are the different mouse models that are used in cancer research. So here you have 4 different types of mouse models. First one is cancer stem cell model, right, where the mouse models are produced by introducing the carcinogens into stem cells, or these stem cells will be injected with some inducers which may generate tumor in the progeny. Right? Then you have clonal evolution model where the cloned organisms are used as a mouse models. In next one, naturally existing model where naturally the mouse will be having the cancer which are selected for the research. Other one is artificially induced models. So here you will be inducing the mouse to acquire or get cancer either by means of radiations, that is physical carcinogens, or by means of chemical carcinogens. Then once these cancer models, right, the cancer mouse models are obtained, so these can be used for further studies in the cancer research. So you can see here the cancer stem cell model and the clonal evolution model are highly used in the studies of cancer heterozygosity. You can know the heterozygosity of that cancerous cells. Whereas the naturally existing model and the artificially induced models are mainly used in studying the biochemical or the pathological studies. Right? Biochemical or genetic pathways, and also in pathology studies or the research, these cells, mouse models can be used. Right? So based on the type of study, you are going to select the mouse model. Okay? Then here coming to the types of cancer. So we said all tumors are not cancerous cells, right? But the cancers are classified. If the tumor is a cancer, then the cancer is classified into 4 types. The first one is carcinoma. The carcinoma, then second one is sarcomas, third one leukemias, fourth one lymphomas, right? So here first one, the carcinomas, where the cancer will start, it will start in the skin or the tissues. So the initiation is in the skin or the tissues, and gradually they spread to the internal organs and glands. They completely cover, the cancerous cells completely cover the internal organs or the glands in the body, right? The next type of cancer is sarcomas. So in this sarcomas, here the tissues which are connecting to the body, right? The connecting tissues of the body such as bones, right? Bones, cartilage or blood vessels. So all the connective tissues will be having this cancerous growth, right? Then next coming to the leukemias. So leukemias, the cancer of white blood cells. Then lymphomas, it is the cancer of lymphatic system. So here these can be in only in particular region the cancer, otherwise it can spread. Like in case of connective tissues of the body, this is highly malignant. That means they can spread from one part of the body to the another part through the connecting tissues, right? It can be a bone cancer or cartilage, or it can be some other cancer which spreads, and it is referred as malignant cancers. Then what is the use? What is the use of these mouse models in the research, cancer research? So the first one is for identifying the carcinogens. Carcinogens are nothing but cancer causing agents. So here in the research, by using the mouse models, you can know what are the agents which are responsible for causing that cancer. It can be a physical agent like radiations, or chemical agents due to the chemicals, or some biological agents, or the genetics, right? Or because of the genes, right? Which gene is responsible for causing that cancer, right? So in that studies it can be used. And then again in case of drug screening. So now once you are inducing the mouse to get the cancer, then what you do? You test the diseased mouse with the novel drugs, right? The novel medicine which is produced by using the pharmaceutical importance. Here the drug screening is done on those mouse, and they check for what is the recovery rate, what is the response of that cancerous mouse model to this particular novel drug, right? So John Hopkins University, they were successful in curing 100% successful in curing the brain cancer in mouse, right? So like that they are testing, so that they act as preclinical trials, right? Before introducing that or before using that on the human, here they are making use of these cancer models. Then next one is in case of cancer therapy, how the mouse is responding to that particular treatment. Then last one is the molecular mechanism of tumor growth and metastasis. So what is the mechanism the tumor is developing inside the body, and how it is spreading, and what is the rate of spread, right? And how it is affecting the other parts of the body, right? So everything, that means all heterozygosity, biochemical and the pathological studies can be done by using these mouse models. Is that clear? If you have any doubts, put it in comments and subscribe for further videos.